So let's take a look at viruses in 11.2. So we're going to look at the structure, and we're going to look at how they replicate, how they cause human disease. Viruses are very strange entities. As we mentioned, they're not cellular. They're not made of cells, and they don't grow and reproduce on their own. They need other cells to do that, living cells. So let's take a look at how they're put together, their structure. So all viruses are made of two types of molecules. They're made of protein, and that's what you're seeing on the outside here. This is protein in the helical type structure, icosahedral structure, or the complex structure. So these are the protein coats we see on the outside, and they look rather alien. On the inside, they either have a section of DNA or they have a section of RNA. Viruses are classified according to their structure. So remember, the capsid shape is the protein coat, an envelope present, and what type of nucleic acid, and do they cause a disease. So here are 4 virus families. There's many more than this, but they're adenovirus, the herpes virus, orthomyxoviruses, and then the coronaviruses. And notice what they have. The coronavirus, a flavor of that, caused the 2020 pandemic, but a regular coronavirus causes the common cold, and it has a section of RNA in it. So viruses are very, very interesting to study. As a matter of fact, a big area of study right now in biology is virology, the study of viruses. Most of us are familiar with the light microscope, the classic biological microscope. However, in the 1920s and 30s, human beings invented a new type of microscope that doesn't use light to see objects, but it actually uses electrons. And the electron microscope was used to see viruses for the first time, and that was in the 1930s. And the virus they looked at they knew there was some entity that was ruining tobacco plants, and the first virus that discovered was the tobacco mosaic virus. And here's a picture of it. Again, you're only looking at the outside of the capsid coat. This tobacco was the first to be discovered. It was seen with an electron microscope in 1935, and you can imagine the biologists' surprise when they saw this wildly geometric shape, and for the first time, human beings discovered viruses. This was 15 years or 17 years after the 1918 influenza pandemic caused by a virus. Human beings had no idea what it was. I'd also like you to take a look down here on this scale. This tiny little scale here says 0.2 microns. So imagine how small these are. Remember, viruses are so tiny that we can't use microns. They're just too small. So we take 1 micron and we divide that by 1,000, and that's called a nanometer. So viruses are normally measured with the nanometer scale. Make sure you read this section of 11.2 carefully, because viruses cause many human diseases. In addition to some that have already been mentioned, viruses can cause rabies, measles, diarrheal diseases, hepatitis, polio, cold sores, and the diseases can range from mild to fatal. Some viruses live in a dormant state inside of our bodies. This is called latency, and don't pop out until later in life. In a disease called shingles, that's what happens there. And some viruses can even cause cancer. HPV causes cancer of the cervix in females. Hepatitis B virus causes cancer of the liver. A viral cancer is likely to develop only after a person has been infected with a virus for many years. Viruses are really, really difficult to control, and one of the sad things is that human beings, up until this point, have not been able to cure any diseases that have been caused by a virus. In fact, what we're able to do is we're able to create vaccines. Vaccines are not a cure, but what they do are vaccines. They train our immune system to recognize the shape of the virus and attack it. And we build up antibodies ahead of time. We have a fascinating, a really, really good animation at the end of this section that I'll have linked for you that talks about how we discovered vaccines. So a vaccine is a substance that contains pathogens such as viruses. Sometimes it's a weakened or a dead form of the virus, but it alerts your immune system so that you can resist the pathogen, the disease causer, and create antibodies to attack it. Vaccines have been produced for many diseases that cause measles, chickenpox, mumps, polio, and several other diseases. Here's a picture of a young man getting a vaccine to prevent a type of viral disease. Viruses are important tools in science research and medicine. As a matter of fact, viral research has increased our understanding of the fundamental processes involved with DNA, RNA, and proteins. And right here in figure 1118, they're using a virus in gene therapy. That's where a normal human gene is inserted into a virus. The virus carries the gene to the human host cell. The gene enters the nucleus and becomes part of the DNA. The normal gene can then be used to make normal proteins. It can also be copied and passed to the normal cells. So gene therapy is a very potent form and studied process very closely in research right now. Let's take a close look at what we've learned in 11.2. So viruses are incredibly tiny particles. Notice they don't call them cells. They're tiny particles, smaller than bacteria or prokaryotic cells. They are not cells, cannot replicate without help, but they have tiny nucleic acids and can evolve. They can change. Viruses can be classified on the basis of the capsid shape, whether it has an envelope, and the type of nucleic acid. Another thing I want to mention is one of the reasons that a new flu vaccine, a new flu shot, is developed every year is because viruses do evolve very, very quickly. Viruses were assumed to exist before they were first seen by an electron microscope in the 1930s. After infecting a host cell, a virus uses the cell's machinery and metabolism to produce more copies of itself. And what it ends up doing is it ends up killing those cells. Viruses cause many human diseases and disturb the homeostasis. They are not affected by antibiotics. The same chemicals, antibiotics, that would kill bacteria have no effect whatsoever on a viral disease. They can be treated with some antiviral drugs or prevented with vaccines. Remember, we don't have a cure yet for any diseases caused by viruses. And viruses can be useful tools in research and in medicine. All right, the last thing I'd like you to do is I'd like you to watch this science animation called How We Conquered the Deadly Smallpox Virus. And they'll also talk about how we get vaccines and how viruses work. So this will be linked if you're watching on the screencast up here in the upper right, but it's also linked in the presentation itself, which you have access to. Good luck. Study 11.1 and 11.2. We'll talk to you soon.